We are investigating the role of FOXG1 and DLX5 in the development of a monogamous species under the guidance of our principal investigator, Dr. Sylvester. Our primary research objective is to examine the role of transcription factors in the developing central nervous system of convict cichlids, to understand how gene expression influences behavior involved in monogamy. We want to understand pair bonding in our model species. Convict cichlids. This fish species display common traits associated with pair bonding, such as mutual defensive shelter, biparental care of offspring, selective partner preference, and selective aggression. Is there a genetic basis for the development of a brain that is wired for complex decision-making and pair bonding? In 2013, Dr. Sylvester demonstrated that during development, differences in strength and timing of gene expression in the forebrain of related cichlid fish species resulted in diverging habitat preference during adulthood. In our case, we are studying how gene expression leads to divergent social behavior. Similar gene expression occurs during development that occurs across animal classes. We are comparing zebrafish, a non-monogamous fish species, to convict cichlids. We want to know why one species forms long-term social bonds while another does not. We chose two genes that code for transcription factors that induce cell differentiation and migration in the telencephalon. FOXG1 is one of the earliest genes expressed in the telencephalon. This figure of zebrafish embryos shows FOXG1 stained in blue on the left and marked in red on the right. DLX5 is a marker of GABA-ergic neurons which connect bundles of neurons in the basal ganglia to the prefrontal cortex. In this image, you can see a zebrafish embryo which shows DLX5 expression in the telencephalon and diencephalon, and you can see characteristic bunny ears that mark DLX5 in the telencephalon. Both transcription factors are involved in the development of the reward pathway. One action of this pathway involves regulation of partner preference due to the influence of dopamine and oxytocin in monogamous species. Prairie voles, a common rodent model of monogamy, are compared often to montane voles, who show different brain structure. Prairie voles have higher oxytocin receptor density in reward-associated areas in the brain. When you inject dopamine receptor antagonists into the brains of prairie voles, there is a reduction in preference of their pair-bonded partner relative to association with a stranger. Zebrafinches are a common bird model for monogamy. Researchers found that when newly paired, male and female zebrafinches had higher levels of dopamine in reward-associated areas than their unpaired counterparts. We want to understand how gene expression influences development of monogamous behaviors. We suspect that interspecies differences in expression of genes FOXG1, a marker of cortical projection neurons, and DLX5, a marker of GABA-ergic neurons, will show differential gene expression during neural development of convict cichlids. To visualize the expression of these genes, we design new primers from degenerate nucleic acid sequences, meaning genes from a closely related species, to the convict cichlids. Then we took RNA polymerase to make RNA-probes, one RNA probe in the 3' to 5' direction, which binds to the complementary mRNA sequence, our target sequence, in the fish embryos. Antibodies conjugated with an enzyme that dye the surrounding domain purple bind to the RNA probes for visualization under a microscope. We perform this process, known as in situ hybridization, in the embryos of convict cichlids and compare them to the images of zebrafish embryos retrieved from a genetic imaging database. Please join us in the conference room to learn more about our experiment and the results of our study, and to ask questions about future directions. Thank you.